That's good.
Okay, over to you.
Thank you.
Yeah. Take light one.
All right, so what? 10:03. All right, good morning. All right, so let's get started again.
Please carry on.
All right, so I understand many of you are interested in drug discovery or structural biology and drug discovery. So I will focus on more, I guess, I will show you our experience working with fatty acid metabolism and how it's linked. So of course, to do drug discovery, you need to understand the target protein first. And structural biology, you see, have a tremendous role in helping you to understand the biochemistry of the enzyme, which then you can turn around and use that to develop molecules against the target. Okay? So, and this is linked to the obesity epidemic. So in the US, of course, it's a major problem. 30% of the population, though this slide is old, 2011, but I don't think things are getting better. Well, maybe over the past couple of years, things are a little bit better, but anyway. So a third of the population is obese, another third is overweight. So a third of the population, adult in the US, have a weight problem. And then it's a problem actually all over the world, in developing countries as well, I think India as well, I guess my understanding is. But being obese is already an issue, but then obesity also predisposes individuals to serious diseases such as diabetes, heart disease, cancer, and other issues. So the whole system is called the metabolic syndrome. So the idea that maybe pharmaceutical intervention in fatty acid metabolism could be a way of controlling body weight and also be helpful or efficacious against the diabetes and other diseases. So that's sort of the general concept here. So that leads to the enzyme that we'll be talking about a lot today, is the acetyl-CoA carboxylase, or ACC. So these are crucial enzymes in fatty acid metabolism. And you all took undergraduate biochemistry, right? So you probably all learned ACC1 converts acetyl-CoA to malonyl-CoA. And that drives fatty acid biosynthesis. And that happens primarily in liver and adipose, lipogenic tissue, liver and adipose tissues. So that you probably all learned. What you may not have learned, actually, there's a second isoform of ACC in humans called ACC2. It does the same biochemistry, converting acetyl-CoA to malonyl-CoA. However, this happens primarily in muscle tissues, heart and skeletal muscles. A little bit in liver as well, but primarily muscle tissues. And this malonyl-CoA is not involved in fatty acid biosynthesis. Instead, it's a potent inhibitor of fatty acid oxidation. So two isoforms, same biochemistry, very different physiological functions or effects on the body. So you can imagine that if you remove ACC2, you remove the inhibitor, you turn on fatty acid oxidation. And that, as you can imagine, could be good because you start burning fat. That's exactly what Sully Walkill's group observed when they produced ACC2 knockout mice back in 2001, published in 2001. So they showed that ACC2 knockout mice have continuous fatty acid oxidation and reduced fat storage. So this is sort of how we got into ACC as for studies because of this paper back in 2001. So the paper was published in 2001. I saw it, I think in 2003. Not right away, but anyway, that's 2002, sorry. And that's when we started working with this system. So let me show you one figure from that paper. So here on the left panel is food intake, how much the mice ate. So knockouts are the filled symbols, wild types are the open symbols. So M means mutant, W means wild type. So you can sort of see mutants are filled, wild type is open. So you can see based on that panel, the knockouts actually are hypophagic. They keep on eating, they can't stop eating. Okay? Because I think the monocyl-CoA that's produced by ACC2 somehow is linked to, is a signal, satiety signal, tell you that you're full, you stop eating. Now you knock out ACC2, they don't have that signal anymore, so they keep on eating compared to the wild type. But then on the flip side, the right panel, if you look at the body weight of the mice, even though the knockouts actually ate more than the wild type, they weigh less from females and males. Oh, sorry, male at the top, female at the bottom. So essentially, it's sort of, if you can joke about it, the best of both worlds. You can eat all you want, you still weigh less. But anyway, so that leaves the idea that maybe the inhibitor against ACC2 maybe control body weight and then be efficacious against obesity and diabetes and other diseases. So that's sort of the idea. So then maybe the inhibitor against ACC2 could be turned into drugs. Whether that compound can also inhibit ACC1 was debated tremendously in the early 2000s because ACC1 is essential. If you knock out ACC1, it's lethal. So there are a lot of concerns about if you inhibit ACC1, it may not be good for the cell or for the organism. Of course, once ACC1 is lethal only during embryonic development. Once you're born, it's no longer lethal because you can also get fatty acids through your food intake. Right? So they're another source of fatty acids for your lunch and breakfast and dinner, everything. And also, if you think about it, these are critical cellular processes for fatty acid biosynthesis and fatty acid oxidation. So the cell will also want to regulate this process very carefully. And it turns out, actually, there's a protein kinase called AMPK, AMP-activated protein kinase, which simultaneously inhibit ACC2 and ACC1 by phosphorylating them. We'll talk about protein kinases on Friday. I'll show you actually AMPK a little bit on Friday. Well, today a little bit as well. But anyway, so AMPK is known as the master metabolic switch that simultaneously phosphorylate ACC2 and ACC1 and turning them both off at the same time. So nature can do it because nature can use the AMPK as the inhibitor of both ACC1 and ACC2. So I think many people also believe that essentially you can't have an isozyme non-selective inhibitor that inhibit both ACC1 and ACC2. Of course, the jury is still out because right now no drug against ACC is on the market yet. Many have gone into clinical trials. I'll show you a couple examples today. So ACCs belong to the family of so-called biotin-dependent carboxylases. Okay, so you all know about biotin as a label for your experiments, right? That's all you know about biotin because it binds streptavidin tremendously that you can use that to pull downs and all that stuff. But biotin has a biochemical function. The biochemical function of biotin is a CO2 carrier, carbon dioxide carrier. So because these are carboxylases, so need CO2 to do the carboxylation. And that CO2 is carried by biotin. So these enzymes have two separate biochemical activities. So acetyl-CoA carboxylase or biotin-dependent enzymes altogether have two separate activities. First, the biotin is shown right here. This is the compound biotin. Okay, biotin is linked to a lysine side chain on a carrier protein, biotin carboxyl carrier protein. I'm sorry about all of these alphabet soup today, all these acronyms. Hopefully I'll try to be slow anyway. So BCCP is a carrier protein that carries the biotin on this arm. So a lysine side chain plus this very long side chain from biotin, and then the tip is the active part of the group. So if you look at it, so if you have this thing, everything fully extended, okay, you have an arm that's about 60 angstroms in length. You can build a model with everything fully extended. It measures the distance from the CR for the lysine to this atom of the biotin is about 60 angstroms. Okay, so I'll come back to that. So the first reaction is you need, as I said, biotin is a CO2 carrier. So you need to put the CO2 onto biotin. And the CO2 goes onto the N1 prime position of the biotin. And that's done through using bicarbonate as a CO2 donor. And then also requires ATP hydrolysis. So ATP is hydrolyzed, and then the hydrolysis ATP basically dehydrates bicarbonate to CO2, which then attacks the biotin, and then the biotin becomes carboxylated. So that's so-called BC, biotin carboxylase. So that's the enzyme that carboxylates the biotin. So basically, this activates the CO2. And then for the second reaction, the biotin translocates to the second active site where the CO2 is transferred to the acceptor molecule. In this case, I drew acetyl-CoA because we're talking about ACC. Yeah, as you see later, that your other substrates can be here as well, besides acetyl-CoA, different biotin carboxylases. So the CO2 goes to acetyl-CoA to make you the monocoid carboxylated substrate. Okay? So that's all good. So that's so-called carboxyl transferase. It transfers the carboxyl group from biotin or carboxybiotin to the substrate. So for a long time, people thought biotin is linked to BCCP through this 16-angstrom arm. And everybody thought that the arm is long enough to go back, the arms start waving back and forth because the biotin needed to go back and forth. So everybody thought this arm moving back and forth is enough for the biotin to reach both the BC and the CT active site. That's so-called swing arm. The arm swings back and forth. The domain stays. But then as I'll show you later, actually, when we have structures of these large enzymes, we found out actually the active site distance between BC and CT is much larger, 40 to 80 angstroms away from each other. So there's no way for something that's 60 angstrom length to reach 80 angstrom distance, because if you go one extreme to the other extreme, you get about 32 angstroms. No way you can reach 80 or even 40 you cannot reach. So now the idea is that the entire domain, that the carrier domain actually goes back and forth. Rather than just the arm swing around, the domain actually swing around as well. I'll show you a movie of that later as well, or some examples of that, essentially. Okay? So you'll hear a lot about BC, CT, and BCCP today. And you'll hear actually a couple more. But anyway, that's set the stage. So these enzymes, as you can imagine, are critical enzymes in cells, so they're conserved from E. coli to us, to humans. So down below is the E. coli enzyme. So you have, actually, it's multi-subunit. So you have the BC subunit, BCCP subunit, and then alpha and beta subunits for the CT activity. So 4 subunits forming this holoenzyme that can do the ACC reaction. Up here is the yeast enzyme, eukaryotic enzyme. So as you can see, it's a multi-domain but single chain. Gigantic enzyme, 2200. As you see soon, human enzyme looks just like the yeast enzyme. Well, we are eukaryotes as well. So these are 250 kilodalton enzymes with domains that are homologous to the bacterial enzymes. BC domain, BCCP domain, and then CT, 2 domains for the CT activity. So these color schemes I'll use all through my presentation this morning. So I guess that's help you figure out where things are. And then the other thing you see, essentially, ACCs have this central region that's sort of unique because bacterial enzyme doesn't have anything here. Okay, you'll see something more about that as well. So how would you inhibit? So we're looking for inhibitors of ACC, right? So how would you kill this enzyme? Well, you all know about active site inhibitors, right? So you can kill the enzyme by inhibiting the CT activity or by inhibiting the BC activity. If you find inhibitors like that, either in the active site or allosterically, you can inhibit them. I'll show you an example of both. And then the middle part of ACC, what does it do? Once we understand why this is there, maybe you have a better way of maybe finding some molecules or whatever that can regulate this part and also use that to inhibit ACC activity. I should also mention that this region here, if you do a blast search looking for sequence homologs in the database, you find only ACC, you find nothing else. So a sequence analysis doesn't tell you what this part does. It's sort of unique to ACC somehow. It's not found in other systems that can tell you, give you some insight as to what this part could be doing. But I'm a structural biologist. That means we look at structures. And it's better to do new structures. So at the time, so I said I saw the paper in 2002. So I said, okay, this is an interesting system to work on. So what can we learn from a structural perspective? What's new we can learn? At that time, in the early 2000s, the structural E. coli BC has been done. The structural E. coli BCCP has been done. But the structural E. coli CT or any of the CT actually had not been done. In addition, we know back then, this is early 2000, working on something that's 250 kilodalton is a pipe dream. There's no way you can get there. So we have to divide and conquer. Basically take this eukaryotic one and divide it up into domains, and hopefully that'll give us crystals, and that'll be, back then, crystallography is still the only way to go. So hopefully we can get the crystal, get a structure. Okay. And that's still the divide conquer technology is still used these days now, still. Even from cryo-EM, sometimes you may want to chop out something that maybe not good for your sample, or this and that. But anyway, so this is early 2000s, so crystallography is the only way to go. And then, as I said, we already know the structure of this part, but there's nothing known about the CT part. So okay, we'll focus on the CT. Now I keep on showing you the yeast enzyme here, because again, back in early 2000, as I mentioned in my first lecture, to make crystallography, you need to make crystals. And before you can make crystals, you have to purify your protein. Before you purify your protein, you have to express your protein. And before you express your protein, you have to have a piece of DNA. And this is a pretty large piece, 800 residues. So for the human, I look for the human cDNA, complementary DNA. I looked for it back then, couldn't find it. So then I can purchase a yeast genome from a little company that sell you prepared genome. Yeast cerevisiae, Saccharomyces cerevisiae, is a beautiful organism in the sense that many of the genes in there have no introns. So the genomic material is the same as the cDNA. You can just PCR it from the genome, you get your gene. yeast ACC has no introns. So we just basically PCR'd this piece from the yeast genome. That's how we got the protein sample. Nowadays, different. You can do probably this by using synthetic DNA. But now we're 20 years later. Anyway, so that's our first structure, published in 2003. We started, as I said, 2002. So this is structure of yeast ACC CT domain, the carboxyl transferase. So left is, you see this quite often. I'll show the 2D view here, and then the 3D movie to help you give you a 3D. So what we see actually, the enzyme actually is a dimer. So molecule 1 on the right, cyan, yellow for the 2 domains. So we found 2 domains in each CT. Well, as you saw, there are 2 subunits in bacteria, corresponding to 2 domains in the yeast ACC. So we call it N domain and C domain, N domain, C domain. And then the second monomer, head to tail, N domain at the bottom, C domain at the top. And we also included acetyl-CoA in the reaction in the crystallization, because acetyl-CoA is a substrate. We like to know where the active site is, where the substrate binds. And it turns out we only observe CoA, acetyl group got hydrolyzed during the crystallization experiment. But CoA is bound, as you can see right here, at the interface between the 2 molecules. The cyan domain, which is the N domain of one monomer, and the green is the C domain of the other monomer. So essentially, the active site is right at the interface between the 2 molecules. So that structure right away shows that the dimer of CT is required for the activity of the enzyme, which is consistent with people with the knowledge back then that we know that ACC needs to be a dimer to be active, and the structure actually shows that exactly why, because without a dimer, the CT active site would not be complete. So that's shown a little bit more here. So here is the CoA that we observed based on our structural analysis. So here's the N domain of one molecule, C domain of the second monomer. So that's why there's a prime on all the labels, because actually it's not the same monomer, a different monomer. And then you have CoA is bound mostly to the N domain of this monomer and reaches right at the interface between the two molecules. And then if you look at the molecular surface in this region, colored by electrons, so red is negative charge, blue is positive charge, you can see there's a huge mountains on both sides surrounding a canyon right in the middle. That's at the interface. CoA binds the bottom part of this canyon. And then you may remember from that introduced you to the biochemistry, this enzyme requires two substrates, acetyl-CoA and carboxybiotin. So acetyl-CoA binds here, as you see later, biotin binds to the top half of this canyon. So this essentially exactly is at the site. Okay, so this sort of tells us how the enzyme works. Substrate binds here, catalysis happens, and it defines also the landscape of the anti-site region. But your interest is small molecules, so here they are. So it turns out actually ACC is a known target for some molecules, especially haloxyphop, I'll just call it the FOP compound, is a known herbicide. So it's used to kill weeds by farmers. It has been used for tens of years already in the field. So and then the DIM compound and then penoxidine are also herbicides. They are a little bit newer, but still they have the same property. They can inhibit, they are potent inhibitors of plant ACC. That's why they kill weeds. But they are much less active against yeast or human ACCs that give you some selectivity. And actually exactly how these compounds select against human or yeast ACC rather than, we still don't fully understand. I should also mention the ACC enzyme is actually highly conserved from plants to yeast to humans. So between yeast and humans, 50% identical in sequence. So it's very, very conserved enzyme. Okay? So these three are well-known inhibitors of ACC. And then this compound here came from Pfizer, the pharmaceutical company in Connecticut. I'll show you more about this. They did a high throughput screen against mammalian or rat ACC, and they found, and then followed by medicinal chemistry, they developed this compound called CP640186. That's a potent inhibitor against mammalian or human ACC. So we have actually obtained structures of each of these compounds bound to CT. They're all CT inhibitors. So we sort of know how they work at the molecular level. So I'll just show you a couple examples, the top two. So first, haloxyphop. So it's a herbicide we can buy very easily. And then first we did this enzymatic assay, which I assume you're all very familiar with, biochemical enzyme kinetics. So that's shown right here. So this is the Lineweaver-Burke double reciprocal plots. Familiar with that? Yes? No? Okay. Remember biochemistry, enzymatics? Oh, okay. All right, anyway, so here on the horizontal axis is monomer-CoA concentration. On the vertical axis is the velocity at increasing concentrations of the inhibitor. And then you can see 0 millimolar, 1 millimolar, 3 millimolar. So very high concentration are used to gain the effect, because based on this titration, we can get the Ki as about 0.2 millimolar. So not very potent against yeast ACC, but certainly the inhibitor against the ACC-CT activity. And also based on the three lines intersecting on the vertical axis, it tells you the compound actually is competitive against the substrate. That's the beauty of doing enzymology. You can tell you whether the compound competes against substrate or not. Okay, so it tells you that the compound competes against monomer-CoA. That means the compound probably binds where monomer-CoA or acetyl-CoA binds, where the substrate binds. So to really show that, we determine the crystal structure bound to the enzyme. So here you have beautiful electron density, again this afternoon, of the compound. observed by crystallography. And then here is where the compound binds. Not too surprisingly, it binds the active site because it competes against the substrate. So the compound in black right here, CoA, as you saw earlier in the earlier slide, earlier structures right here. So they are actually bound to roughly the same spot. They compete against each other. That's sort of how the compound inhibits the enzyme. But this actually is only the big picture story. But in detail, actually, it's very complicated. It turns out for the compound to bind, at least to East ACC, it requires a very large conformational change in the CT dimer for the compound to bind. So what I show you here is the overlay between two structures schematized tremendously, only focusing on the part that show large changes. So here in gray, so the overlay two different structures. In gray is a free enzyme. In color is the enzyme bound to the compound, haloxyphosphate, which is shown right here. So you can see in the free enzyme, you have a tyrosine, you have a phenylalanine facing each other. And they touch each other, and actually, that's shown right here. This tyrosine, 1738, is right here. This phenylalanine right there. So they are facing each other. So they are basically covering up the dimer interface. This is essentially, this came from one monomer, this is the other monomer, so that's the dimer interface. So before the compound can bind, this tyrosine had to assume a different rotamer. You remember rotamers? Okay, so the tyrosine changed the conformation. In addition, the phenylalanine also assumed a different rotamer. Going from here, you can see the side chain changed. In addition, there's a 2.5, oops, wrong direction. There's a 2.5 degree rotation of this monomer relative to this one, so there's also the main chain of this phenylalanine moved back by 2 angstroms. So as a result of that, you see the consequence. So in the free enzyme, the dimer interface is completely covered. There's no opening, no anything. Because of this conformational change, a pocket opened up. And then the compound is inserted into this pocket, which is the dimer interface between two monomers of CT. And if you look at the chemical structure of haloxyphosphate, this part is actually quite hydrophobic. Only the tip contains carboxylate, so that's hydrophilic. So the hydrophobic part is inserted into this pocket, which is the dimer interface, which you can imagine is very, very hydrophobic. So some other people found that there's a natural product called sorphin A. That's a potent inhibitor against eukaryotic, including human BC activity, the BC domain, the internal half. So you can see this is the compound here, sorphin A, made by a Serrangium bacterium. And the polyketide, 18-member microcycle, and 1 nanomolar Kd, as I mentioned. So it's made by bacterium to kill fungus. So of course it doesn't kill bacteria because it doesn't want to kill itself. So it's selected against eukaryotic enzyme, but not active against bacterial enzymes. So how does it work? So let me tell you the story. Oh, let me go back. So this actually was used as a target for developing essentially herbicides by a company in North Carolina in the US using this compound as a possible lead molecule as a herbicide. So, and they realized, since we published by then structures of ACC and stuff, they thought I'm an expert on ACC. So they reached out to me, this company reached out to me and said, we have this compound, we'd like to know how it works. Can we collaborate? I said, sure, why not? I want to learn everything. So that's what we did. We collaborated with the company and we determined the structure. So we purified yeast BC again, yeast because we can get the template by PCR from the genome. So we expressed purified protein, determined the structure. Here the structure right here. So a couple of surprises. First, CT, as you saw, is a dimer, right? The anti-sided dimer interface. BC, when we determine the structure, actually is a monomer. It's not dimer. In addition, BC somehow is inactive as a monomer. So again, consistent idea that ACC needed to dimerize to be active, but we don't understand why BC is the monomer on its own, and we don't understand why it's the monomer. Secondly, remember BC requires ATP hydrolysis. ATP binds up here. And that's the domain open and closed during catalysis. But anyway, so this will be the active site because that's where ATP binds. And sorphin in green binds to the opposite face of the molecule. So this actually is an allosteric inhibitor. So then the question is, how does binding sorphin here affect catalysis up there? We don't have a clue back in 2004 when we determined the structure. So now we do, so I'll get there sooner or later. Anyway, so this is sort of the issue. Why BC is the monomer? Why is not active? How does sorphin binding here control catalysis up there? Now, if you look at how sorphin A is bound, you can see it sort of lies over the surface of BC domain right here. And then there's the deeper part of the pocket right here. So looking at the side, most of sorphin is flat right here. But a little bit here, which is right here, is a little bit deeper. It goes into the sub-pocket. So this sub-pocket actually is quite important. I'll come back to that. So that actually leads to developing a series of compounds. So, and then this phenyl ring here is inserted into the pocket right there. So we know exactly how everything binds, very good potency, but with the biochemistry, we don't understand. So if you look at how the detail of the binding, so you can sort of analyze everything. If you're good at PyMol or CUT, you can draw all these things out. You can sort of see how everything interacts. Unpaired interaction between this glutamate and this arginine, and this serine, this lysine forming unpaired with the glutamate. And the serine is in red because it is known that mutation of serine to a tyrosine confer resistance to this compound. So yeast with the mutation here can survive sorphin treatment. They don't die anymore. And you can look at the structure, this serine sits directly between sorphin. So if you change the serine to a tyrosine, steric hindrance, pocket is gone. It doesn't inhibit anymore. So it makes perfect sense. And then secondly, lysine, this lysine 73 changing to arginine also is known to confer resistance to sorphin. And the lysine sits right next to sorphin, also involved in the unpair, so could be important as well. And then there are other residues in the binding site, for example, this arginine. So we can basically, to prove this binding mode, we can make mutations of residues in the binding site and then do a binding assay. So that's how we did. How do we do a binding assay? So again, structure is informative. The structure shows that the tryptophan right here, 487, sitting right next to the compound. So if you know, well, either you know or you don't know, either way, the hydrophobic group right here from sulfan bind next to a tryptophan. Oftentimes it leads to enhancement of the fluorescence. Tryptophan is fluorescent, right? So because the indole ring here is fluorescent, so the binding of the compound leads to enhancement of tryptophan fluorescence. So you can monitor tryptophan fluorescence as a binding signal. And that's exactly what we did. So we put in more and more sulfan compound into the enzyme. And in the wild type enzyme, you can see a change in fluorescence, enhancement in fluorescence of the tryptophan. And from that, you can get the KD. But you can see based on this, less than 10 nanomolar. And then if you make mutations, for example, this lysine 73 to arginine, you have a tremendous change in, or loss in potency, nearly 1,000-fold. And then if you change this glutamate right here to arginine, you also have maybe 100-fold loss in potency. So this shows essentially when you mess around with the binding site, you lose activity. So confirming that this very likely, this is the binding site for the sulfur molecule. So then the question is, how does it inhibit? So back in 2004, the best thing we could come up with was this idea that sulfur A may be a protein-protein interaction inhibitor. So let me walk you through the thought process a little bit first. So this is the yeast ABC. So I told you the E. coli ABC subunit structure has been done, and that actually is a dimer shown right here. So one subunit is cyan, one subunit yellow, and the active site, ATP binding sites here and here. And it is believed that E. coli BC has to be a dimer to be active. John Cronin's group, many years ago, showed that if they knock out one active site, leave the other one wild type, the enzyme is dead. Somehow that they believe that long distance communication between two active sites, so both need to be active before the enzyme can do turnover. Why that's the case, we don't really understand. And we know, as I mentioned, eukaryotic ACC is a function of oligomers, dimers at least, but then the BC domain alone actually is the monomer in solution, as I showed you based on the crystal structure. But then if you overlay the structure of each species domain, monomer, onto each of these two monomers of the E. coli BC subunit to force a dimer, if you will, sulfan A sits in the dimer interface. So that gave us an idea maybe sulfan A binding here will be, well, we know it will be incompatible with dimerization. So maybe sulfan A basically blocks the dimerization, which is not 100% true because we have the structure of each BC domain alone. That's a monomer as well. You don't need sulfan binding to be monomeric, but sulfan A may stabilize the monomeric form. So anyways, how do you show that? How do you show sulfan A block binding? That's very hard to do because the thing is the monomer already. How do you show that something blocks dimerization on a system that's already monomeric? The best experiment we can come up with is using a native gel. So run a native gel of E. coli, yeast BC domain, wild type BC domain in the presence of more and more sulfan, no sulfan, half sulfan, 1-to-1 molar ratio, 1 to 1.5. You can see when you put in more and more sulfan, BC domain becomes really very sharp, runs very sharp on the native gel, which means it's really stabilized, have a very unique conformation. So it runs very sharp, very fast on the native gel. And then the K73R mutant, as I showed you, doesn't bind sulfan very well. When we put in more and more sulfan, it stays in this larger aggregate. So that suggests that sulfan A certainly does something to the enzyme, to the BC domain. Exactly how, we don't really know because this is only a native gel. It's not a molecular level thing. So we have to go beyond the structural BC domain alone. But before that, so I remember I showed you earlier that, well, first, because Sally kills paper, So the 2000s, almost every company in the US working on ACC as a drug discovery program. So Abbott also did high throughput screen. They obviously have a different compound library, so they found different kinds of compounds. So, again, quite potent, 1 micromolar and 80 nanomolar against ACG1, ACG2. And by doing some medicinal chemistry, they evolved this compound, which is actually very selective, 1,000-fold selective for ACG2 against ACG1. Remember that earlier I said it's not known whether the compound can be targeting ACG1 and ACG2, so this is a compound that targets only ACG2. And then they showed that the compound actually more effective at reducing mononuclear levels in liver tissue rather than muscle tissue. So that's consistent with the ACG2 selectivity. But how far this compound advanced in terms of drug discovery, we don't really know. This is so there are publications back in 2006 when the company decided to stop the metabolic disease program. So we don't know what happened afterwards. From us, so I said, as I showed you, we determined the structure of BC domain bound to sorphin A. We determined the structure of CT domain bound to very different ligands. The real understanding of this enzyme will be the structure of the entire thing, which we call the holy grail in the lab, because we started back in 2002 or 2003. So we struggled with this for many, many years. And this is sort of one thing about scientific research. You can struggle with something for a long time. That's true. You keep on struggling. If it's important, you keep on doing it. At the same time, if you cannot make progress on this direct target, you look sideways on something that's related. And that's shown right here. So biotin-dependent enzymes, I told you ACG1, ACG2. Actually, there are a total of 5 such enzymes in humans. So these are the human ACG1, ACG2. Look just like the yeast enzyme. And there's also PCC. PCC use propionyl-CoA rather than acetyl-CoA, just 1 carbon longer as a substrate. And then MCC is methylcronyl-CoA, a different kind of substrate. You can see they have 2 subunits, alpha and beta. Alpha looks like the N-terminal 1/3 of ACC. Beta looks like C-terminal 1/3 of ACC. Doesn't have the middle region. Okay? And then the 5th enzyme called PC, pyruvate carboxylase. So in this case, the carboxyl receptor is pyruvate, not a CoA ester. So as a result, it has the same BC domain, but CT is entirely different because this CT recognizes CoA esters. This CT recognizes pyruvate. So it's a completely different kind of enzyme. So if I have time in the second session, I'll tell you about PC. But anyway, first, what do they do? So here is metabolism simplified. So here's mitochondria. Glucose up here. Glycolysis gives you pyruvate. Pyruvate enters the mitochondria through pyruvate dehydrogenase, which converts pyruvate into acetyl-CoA. And then PC will convert pyruvate into oxaloacetate, and that is part of the TCA cycle, which cycles around. Okay? And then citrate is part of the TCA cycle. We can leave the mitochondria and gets converted to acetyl-CoA. I'll talk about this. I'll talk about that. And this acetyl-CoA is a substrate for ACG1, which make mononuclear-CoA, make palmitoyl-CoA. ACG2 make mononuclear-CoA, inhibit the transport of palmitoyl-CoA into the mitochondria for beta oxidation. That's fatty oxidation. So that's how it works. ACG1 drives biosynthesis. ACG2 actually inhibit the transport of fatty acid from the cytoplasm into the mitochondria. That's how I inhibit fatty acid oxidation. So PC convert pyruvate to oxaloacetate, which also, I'll just show you later, is involved in gluconeogenesis, synthesizing glucose from oxaloacetate. And then PCC and MCC are involved in amino acid metabolism in the mitochondria. So in the case of PCC, it's involved valine, threonine, isoleucine, methionine. When they are in the metabolized, require PCC to convert palmitoyl-CoA ultimately into succinyl-CoA so that it can go into the cycle and be metabolized. Similarly, MCC is involved in leucine metabolism. Same thing, converting that ultimately to acetyl-CoA, then that can enter the TCA cycle. So these are critical enzymes in human metabolism. So there are many disease-causing mutations in MCC, PCC, and PC. So essentially, if infants are born with mutations in these genes, actually it can be a very serious metabolic disease. Okay, so we said, okay, we can't do much with this enzyme because it's so big. Maybe we can work on this enzyme. It's only 700 residues and 500 residues, much easier to do. Well, not really. They are alpha-6, beta-6 dodecamers. So this is a dimer, 400 kilodalton. This is dodecamer, 750, actually even bigger. At least the pieces are easier to make. And this also has bacterial homologs. So we work on a bacterial homolog because, again, we can get the DNA from the bacterial genome. So by screening through many things, I'll skip over all the structural biology, all the painful things we did. So we determined the structure of holoenzyme PCC from a bacterium. That was 2010. So this is the holoenzyme alpha-6, beta-6 dodecamer. We also determined the structure of MCC, bacterial MCC from Pseudomonas. So the two enzymes have the similar, identical, if you say, domain organization, but you can see, look at the overall structure, actually it's very, very different, right? The overall shape is very, very different. And then we also, and they are actually also highly homologous in terms of sequence, 40%, 30% identical. We also look at the database. We found another protein, 120 kilodalton or so monomer, is annotated as a pyruvate carboxylase, but then when we look at it, it cannot be pyruvate carboxylase because the CT part looks like the CT in ACC, so it has to be a CoA-dependent enzyme. And we purified this enzyme, turned out to be a hexamer, 700 kilodalton, just like these. So we determined that structure, looks like that. Again, very, very different. So these are all very similar structures, or very similar domains forming these holoenzymes, but the overall structure of the enzyme actually is very, very different. So I showed you earlier on Monday sequence determined structure, right? You may remember that. Here you have very similar sequences, but you have very, very different structures. So how do you reconcile the two? The way I reconcile them is the sequences of domains are similar, which means the structures of domains are similar. But how the domains are put together to give you the overall structure is very different depending on the enzyme. So I guess Legos are here in India as well. Like when you grow up, you play Legos, right? Building blocks. So you can sort of think about each one of this as a Lego block. You can use the same bunch of Lego blocks to build very, very different things. And that's what nature is doing. Using same blocks, same domains, same collection domains, but put them together differently to give you very different overall structures. So once we have these overall structures, we can measure the distance between the BC and the CT active sites. It's 55 angstrom for PCC, 80 angstrom for MCC, 40 angstrom for, well, LCC. I won't explain why it's called LCC. So this tells you, as I showed you earlier, the distance is very long for the biotin to go back and forth. So biotin is sitting on BCCP, which is this blue domain right here. Another thing is from these structures, from the structural holoenzyme, we actually discover a new domain, which is colored orange, which I named it BT domain, because as you can see, it mediates interaction between BC and CT. So BC, CT, BT. Where does it go? It's actually in this little daylight between BC and BCCP. So traditionally, based on biochemistry, we know we need BC activity, we need BCCP to carry the biotin. And there's a little linker between the two domains. People just ignored it. They say, who cares? Nothing important biochemically, so we don't know what it does. So they ignored it. But then when we determine the structure, we realized this linker here actually forms a domain. So that means now there's a new domain, BC, BT, and BCCP. So what does BT look like? Very simple structure. I showed you yesterday, 8-stranded up-down beta barrel, which is right here. That's another example. That's why, I didn't plan it that way. But anyway, you have an 8-stranded up-down beta barrel. But the surprise here is, at least surprise to me, you have an alpha helix stuck in the middle of it. That was completely unexpected. So back then, the best we can do is secondary structure prediction. Nowadays, you can plug into the AlphaFold, see what happens. Back then, you don't have that luxury. So we'll do a secondary structure prediction. So when you look at the secondary structure prediction for this little segment right here, you have a long helix, you have 1, 2, 3, 4, 5, 6, 7, 8 beta strands, and a little helix. So you could maybe make a model of an 8-stranded beta barrel using these 8 strands. But I don't think back then you had the courage to put the alpha helix in the middle of the barrel. This, as far as I know, the only time I have seen this, something with a helix stuck in the middle of the beta barrel. So long helix is this one here. You have the little helix right here is that one right there. So the secondary structure prediction actually is perfect. So now if you do AlphaFold prediction, chances are you'll get something like this as well. But anyway, so that's the BT domain. So we learned quite a bit about this enzyme, but ultimately that's the holy grail. So why is it so difficult? As I said, 250 kilodalton monomer, dimer is 500 kilodalton, and then this central region I call no man's land because we have no idea what it does. As I said, it has no homologs in the sequence database. In addition, I mentioned earlier AMPK regulates both enzymes at the same time by phosphorylating them. So AMPK put the phosphate here and the phosphate here. So neither is in the conserved region of the enzyme. So you can imagine how does putting a phosphate here can inhibit the enzyme. More importantly, how can you put the phosphate in the middle of nowhere? How does that inhibit the enzyme? No idea. So we need the structure of the holoenzyme. So how the process happened, we determined the BC and CT first, I showed you. And then my postdoc actually finally determined the structure of central region, the no man's land. That's the structure right here. So we have a BT, this piece right here, BCP in blue. And the entire central region, the white part, we found out I had 5 domains here. We call them AC, ACC central domain 1, 2, 3, 4, and 5. So that part has 5 domains. And finally, we said, okay, well, let's just try to express this 250 kilodalton protein E. coli, see whether it works. And it worked. We got half a mega protein for 1 liter of culture. We got enough. Even more amazingly, we crystallized it. So we got crystals of this enzyme purified and expressed in E. coli. And this is all done before the cryo-EM resolution revolution. Something this big we could have done by cryo-EM. Well, not really, as I'll show you soon. So we had to do a crystal. So luckily we got crystals. So it took us about 13 years to get the thing done. So we started in 2003 or 2002. By the time this holoenzyme structure was published was 2015. So the first structure of ACC dimer, shown right here. The whole structure looks like a quarter of a pie. So at the bottom is the CT dimer right here. You can see each monomer has 10 domains. And then as it swings around, you can see at the top. So one monomer as a surface, the other monomer is a schematic drawing. You can see BC now actually dimerizes. So BC now in the holoenzyme actually is a dimer. And then the central region on the left, on the right, they are far away from the enzyme active site. So they are not really involved in the enzyme catalysis, they are just scaffolds. Here is the BT domain. And then the distance between the BC active site and CT active site is about 80 angstroms. Again, you need the entire BCGP to go back and forth to do catalysis. And then BCGP in this case actually sits in the CT active site. Okay, so we can show you a lot more. I can show you a lot more about this. But the main question now is how the BC dimerize? Because I showed you the structure we had earlier, BC domain alone is a monomer. Somehow in the holoenzyme, BC dimerized. So here is a morphing wheel. If you don't know how to do that in PyMol, I'll show you how to do that in PyMol. So morphing between two states. Gray is the structure of BC alone, bound to sorafin. And red is the structure of BC in the holoenzyme. So this is the monomer state. Red is the dimer state. You can see tremendous changes in the dimer interface between the two states. This beta strand moved by a huge distance between the two. And this tryptophan, you remember that sitting next to the sorafin, it swings in and goes into the sorafin A binding site. So in the dimer state, sorafin A cannot bind. There is no binding site. It only exists in the monomer state. Okay, so these large confidence changes in the dimer interface allow BC domain to dimerize. And in addition, we see large confidence changes in the active site of BC. So BC requires ATP and biotin. Biotin is right here. So it turns out in the BC enzyme alone, a loop of BC sits where biotin is. That's how it's inactive, because it blocks biotin binding. And then in the holoenzyme, the loop moves away, biotin can bind, and everything can do catalysis. So in addition, we saw, so a group in South Korea showed actually, remember I showed you AMPK put the phosphate just before the BC domain. That phosphate is right here. So that phosphate is recognized by the same pocket that binds sorafin A. So basically, the phosphorylated BC domain can only bind to the monomeric state of the enzyme. So combining all of these different information, we have this model right here. So BC is a dimer where it's catalytic active. And dimer can dissociate into monomers, which also will be active. So this means biotin right here. And then interestingly, in the case of yeast enzyme or human enzyme, the dimer interface undergoes a very large confidence change. And this state can no longer dimerize. In addition, coupled with this confidence change in the dimer interface, there's also a change in the active site. A loop goes into the active site, kick out biotin. So now this state will be catalytically inactive. And what sorafin A does is sorafin A binds to this state, stabilizes this state. Similarly, phosphorylation at the N-terminus of BC domain also binds here to stabilize it. So that's how they stabilize it. That's how sorafin A and phosphorylation inhibit the enzyme by stabilizing this inactive monomeric state of the enzyme. So having the structure of the holoenzyme allows us to understand how sorafin A works at the molecular level. Basically, mass transport, so the active conformation is converted to inactive conformation. Okay? So coming back to sorafin A. So I showed you very potent compound. So a small biotech company called Nimbus became interested in BC as a drug discovery target. So they looked at the binding mode of sorafin A to human ACC. They recognized this pocket right there. So they started doing in silico screening. Remember high throughput screening? Those are physical screening. You actually use actual compound to screen against the actual enzyme. Here you screen using computers. You predict the binding mode of the compounds and predict the binding affinity, and then you rank all of the compounds. So the idea is by doing so, you concentrate the likely active compound near the top. You acquire a few of those and then see whether they are active or not. So by doing this, they found a lead compound that has micromolar affinity or activity against BC activity or ACC activity. So now this is all so far computational and biochemical assays. So they want to know how exactly the compound works at the molecular level. Again, they realize I'm an expert on ACC, so they reach out to me and said, okay, can you help us determine the structure of their lead compound? I said, oh, of course, I'd like to show you wrong because this is in silico docking. How close can that be? So let me show you. So this is their the silico docking results. The pose on the silico docking, this is their compound. Basically using this pocket right there, and then keeping the phenyl ring. And then the rest of sulfone A is ignored. So the compound basically binds right here, as you see right here. And then the similar compound we determined using crystallography, and that's shown in cyan, coming out right here. And I was shocked how close they are. So this is back in 10 years ago. Okay, it's not now, 10 years ago, more than 10 years ago. So you can see the binding mode is actually quite similar between what's predicted by in silico docking, completely computational, versus what happens in actual crystal. The biggest difference you see is actually this ester group here. The carbonyl is out of the plane of this aromatic group. In the crystal structure, you can see the carbonyl actually in plane. The ester is actually in plane. The whole thing actually is a very large pi system. But overall, actually, it's very, very similar. This goes to show you the power of computational chemistry. And this is 10 years ago. And now, of course, it's even more powerful. In silico docking and everything else is used even more commonly nowadays to find either leads directly or at least help you to concentrate compounds that are more likely to be active, so you don't have to screen through a million compounds. Now you can only screen through a few thousand or maybe 10,000 compounds. Much easier to do experimentally. So based on this knowledge, they did additional chemistry. They found this compound called ND630, a very potent 1 nanomolar against ACC, and this essentially became a lead molecule. So the compound inhibits fatty acid biosynthesis, stimulates fatty acid oxidation. Again, isozyme non-selective, inhibit both isoforms of ACC. And then that's in cells. And then in animal models, in rats, reduce liver muscle melanocholine levels, reduces hepatic steatosis. In rat model, diet-induced obesity, insulin sensitivity, reduced weight gain. Again, all the right hallmarks that you like to have for a lead molecule. So this slide is kind of old. So this actually entered phase 2 clinical trials against NASH, which is non-alcoholic steatohepatitis. So it's a very serious, very difficult liver disease. And he entered phase 2 trial against liver disease because Gilead stepped in and bought the company back in 2016. So when that paper came out in PNS, that also helped essentially the whole process. And then so in April, Gilead basically put in nearly 1.2 billion, essentially 400 million upfront, 800 million based on milestone payments. Okay. So I don't know exactly where the compound is right now in the clinic, but Gilead, of course, has a long history of developing or treating liver diseases. So that's why they're interested in this drug discovery aspect. Okay, so this is not the story that you can sort of see. You can get a compound and then go from there to a drug lead into the clinic. Okay, so as a little side, I know the next slide is not on your printout, but anyway, it's supposed to be a joke. Hopefully you can understand it. So structure-based drug discovery, I'll show you a few examples now. Used to be called rational drug discovery. You're probably too young to know that. But anyway, let me tell you, when I was at your age, well, older than your age now even. Anyway, so, but then the people who did all the designing are the chemists. They didn't like the term rational drug discovery because to them that means what they were doing beforehand was irrational. And they don't like to be called irrational. I can understand that. So a new term needs to be developed. So back in 1995 or 1996, whatever, so a well-known journal came up with a new term. What do you think? Is that better? That means what they did before was, I won't say it. Anyway. But this is actually the more important concept. So as you see, it's much easier to develop a potent inhibitor, maybe selective as well. You can get to nanomolar affinity in the academic setting quite easily, but it's much more difficult to get a drug out of that because there are many other properties the molecule has to have before it can become a drug. And that includes, if for most small molecule compounds, you like to take them orally as a pill rather than as an injection. And so oral bioavailability is a critical issue. Many compounds are very good inhibitors, but they're not bioavailable. That means essentially, if you take it through the mouth, does it go into the bloodstream? If it doesn't go into the bloodstream, it benefits the toilet, doesn't benefit your person. So it doesn't help. Also, low toxicity, efficacy, and finally, of course, these are all developed by companies. So in the end, they like to make money out of it. So marketability is another critical issue as well, whether they can make money out of it. Of course, nowadays you have a compound that costs $300,000 a year to treat the patient. So even for small diseases, sometimes you can still have drugs being developed for them. Okay, so last story about ACC is AMPK phosphorylation. So I showed you already how AMPK phosphorylation here can inhibit the enzyme, same as sorafenib A. So that's the same. So the question is, how do you put the phosphate here and phosphorylate and then kill the enzyme? In addition to that, it turns out yeast ACC doesn't have a site here. That serine is not there. I'll show you on the next slide. The suspicion is the yeast ACC is phosphorylated in the middle as well, just like the human enzyme, but exactly where was not known. And the yeast AMPK is known as SNP1 because of how it was discovered. So here is a sequence alignment. So the end-terminal phosphorylation site, ACC2, the serine is right here, ACC1 is serine. Yeast ACC has an isoleucine, so it cannot be phosphorylated there. In the middle region, no man's land, human ACC has serines here, so they can be phosphorylated. Yeast ACC 1157 is a serine. So the suspicion is that this 1157 serine is the one that's phosphorylated by AMPK or SNP1. But there's no direct evidence for that either. So where is this 1157? We have the structure of each holoenzyme, so we should know where it is. It's right here, in the disorder loop in this domain here. So you can see it's in the scaffold, of course, no man's land, 75 angstrom away from the CT at the site. So how can you put the phosphate in here and inhibit the enzyme? More importantly, 1157 is in the disorder loop of domain AC4. So you can see 1136, 1170. Between them, we don't even see in our crystal structure. So that loop is flexible. So how can you put the phosphate in a flexible loop and then somehow inhibit catalysis by this enzyme? So we'll have to understand how that works. Once we understand how that works, maybe you can we know if you mess around here, you can kill the enzyme. So if you understand that, maybe you can find compound that can do that as well. So first, we developed an in vitro phosphorylation assay. SNP1, just like AMPK, as I'll show you on Friday, is a heterotrimer. 3 proteins that we express and purified. And it's a protein kinase. It needs to be activated by another protein kinase through phosphorylation. So TAS3 is upstream kinase. So when we purify that, treat SNP1, we get phosphorylated or activated SNP1. And that can phosphorylate ACC. So we use the AC3, AC5 sample, 3 domains only from the middle region. You can treat that with everything, SNP1, TAS3, the substrate, and ATP. You can sort of see it become phosphorylated. The way you tell that, because the phosphorylated protein run a little bit slower on the SDHL. You can see essentially almost 100% conversion between the unphosphorylated form to the phosphorylated form. And using this, we can do biochemical assays. So you can see the wild-type enzyme without TAS3 is active over 30 minutes, no problem. It's fine, it stays active. But once you add TAS3, which phosphorylates SNP1, which in turn phosphorylates ACC, within 10 minutes, the enzyme is dead. So we know that. So now we make a mutation of that serine to alanine. Over 80 minutes, in the present top 3, you don't see any decrease in activity. So that means that serine very likely is the only phosphorylation site in East ACC. So using that in vitro phosphorylation system, we purified a fully phosphorylated sample, crystallized it, determined the crystal structure, shows how the phosphate on serine 1157 is recognized by this serine, this tyrosine, and 2 arginines. So once we know how the serine is recognized, we mutate the 2 arginine to alanine. Now we show that essentially now this mutant is no longer sensitive to phosphorylation, confirming this binding site. So that shows essentially this 1157 certainly is phosphorylated, and then recognized by these 2 arginine side chains in the protein. Okay, so how much time do I have? We have until 12, is that right? I think I'm good. Okay, so now how does it work at the molecular level? Of course, allosteric inhibitor, you think conformational change. Something changed in the enzyme. So we compare the structure of the AC3, AC5, the 3 domains alone, which is shown in the light color here, versus the phosphorylated version, which had the phosphate right there. We were expecting some conformational difference, but you can see they are identical. No difference at all. I said, how could that be? Luckily, we had the structure of the holoenzyme. In the holoenzyme, you can see AC3, AC5 alone, actually structure is very, very different from the structure of AC5, 3AC5 in the holoenzyme. If you overlay the top part, AC5 has a 40-degree rotation. So dramatic conformational changes in this middle region between when you cut it out, it's one state. In the holoenzyme, it's a different state. So it's very dynamic structures going back and forth. And that's how essentially the enzyme can form this holoenzyme. So this is 2016, somewhere around there now. So we heard by then that EM has become very powerful, and our enzyme is 500 kilodalton. So it's a beautiful sample for doing cryo-EM. We said, well, let's look at it by EM, see what it looks like. So we put our sample in anti-stain EM. This is not cryo-EM, basically using anti-stain to see the overall shape of the molecule. You can see actually it's very heterogeneous. You have these curved shapes, elongated shapes, little bit of globular shape. You can all kind of shapes in here. It's a huge mixture, conformationally highly heterogeneous. But this is the exact sample that gave us crystals. As I told you on Monday, in the crystal, everything, all the molecules are the same. How can something like this give us crystals? It's almost impossible. So I started thinking, how did we even ever get crystals? So I thought maybe the trick is the crystallization solution somehow stabilize the sample. So this is ACC in the regular protein buffer. So I told my postdoc, okay, let's try look at EM of the sample in the same buffer that gave us crystals. And that's what we saw. You see a difference between the two? Obviously. Now you have globular things, very much more homogeneous. And then by looking through the crystallization buffer, we realize it's actually citrate. So basically, you put in citrate, you go from here to here. If you don't have citrate, it looks like this. If you have citrate, you have that. So that tells us that even if we had done EM first, we probably see this, it'll never work. And only when we got crystals, we realized we can make it work by putting in citrate. So that came from screening through many, many conditions. So that also tells you the importance of using the right buffer condition to do your structural studies so that your sample is happy under that specific buffer condition. Anyway, using this anti-stain image, you can do 2D classification, get 2D classes, and then you can easily dock in the shape of the overall holoenzyme. In addition, post one step further using these native stain images. It created a 3D reconstruction at about 8 angstrom resolutions, blobs. But still, the crystal structure can be docked in very easily, so the overall shape of this dimer by native stain EM fits the crystal structure very nicely. So we are seeing the same thing in EM and in crystallography in the presence of citrate. And that's the trick. So now we know how to make the enzyme happy. So can we show how phosphorylation works? So if you phosphorylate serine 1157 and look at EM, even in the presence of citrate, you see the dimer fell apart. You see the linear state or the curved state. You don't see the globular state. Okay. Similarly, sulfon A does the same thing. So that shows essentially how they work. So finally, this is the model. To show you the model, first schematize the structure into CT dimer, the central region, and the BC dimer. Okay, so this is how it's schematized. So this is the whole enzyme. It's active. And then if you phosphorylate serine 1157, there's a large conformal change in the central region. As a result, BC domain dimer fell apart, and the enzyme is inactive. So essentially, in this dimer, CT is a strong dimer, it's stable. BC dimer is the weakest link. It's very unstable. It's very easily fall apart depending on what you do to the enzyme. So if you phosphorylate the middle here, which is right here, you cause a large conformal change, and then the BC dimer fall apart. Once the dimer fall apart, I showed you earlier the conformal change in the BC dimer interface and also the active site, so the enzyme is inactive. You get either this elongated form or this curved bent shape, but both are same, essentially. And then the top part is the same what you saw earlier. BC dimer fall apart, either sulfon A or phosphorylation at the N-terminal end of the BC stabilize the monomer form. So you have essentially the inactive form. So in the EM image, in the presence of sulfon A or phosphorylation, you see these states. And then in the presence of citrate with nothing else, you see this state. So that's essentially how phosphorylation and sulfon A regulates the computational dynamics of this enzyme, going from the active state to the inactive state. Okay? So it's a long story over 10, 15 years. I think now we have a very good understanding of how this enzyme works at the molecular level, how the small molecules, how post-transcriptional modification works to regulate the activity of this enzyme. And also how this information can be used for driving drug discovery, essentially. Okay, so the summary, I already mentioned all of that, so I'll skip over that. Moving on to the last topic today, or this morning. So this you saw earlier, but I got rid of most of the PCC, the MECC in the middle of mitochondria because I want to focus on the next aspect, which is citrate leaving the mitochondria gets converted to acetyl-CoA. Acetyl-CoA is critical for fatty acid biosynthesis, fatty acid oxidation. In addition, it's also critical for other processes in the cell, making cholesterol, making isoprenoids, also, of course, acetylation, because acetyl-CoA is the donor of the acetyl group for acetylation. So it's a very important compound in the cell. How do you make acetyl-CoA? In the cytoplasm, in the mitochondria, it came from pyruvate dehydrogenase. In the cytoplasm, it comes from citrate. And citrate goes to acetyl-CoA by an enzyme called ACLY, ATP citrate lyase. So that's the enzyme that does that. So you could imagine, you could sort of say that ACLY is upstream of ACC1, ACC2, because that produces the substrate for ACC. So what is the ACLY? ACLY can convert, as you saw on the previous slide, convert citrate and CoA into acetyl-CoA oxaloacid. So that's the biochemistry. And the reaction actually is fairly complicated. You require 4 steps And the enzyme is very large, 1,100 residues for the human enzyme. So, and also it's a tetramer, so it's about half a megadalton again. But as you see, perfect for EM. So the 4 steps, first require ATP hydrolysis. The phosphate goes on to histidine. So we have a phosphohistidine. And then the phosphohistidine the phosphate is transferred to the citrate group to make citrophosphate. So that's how you activate citrate. And the citrophosphate is then combined with CoA to make citro-CoA. And then citro-CoA can go a lyase reaction to give you acetyl-CoA and oxalic acetate. Okay? So if you look at the enzyme, it has many, many domains, and the domains have sort of known functions. So, for example, the red part is called ATP GRAS domain, so it binds ATP. The cyan part is the citrate domain, binds citrate. The blue part binds CoA, the blue domain. And then the pink domain contains the histidine, which is phosphorylated. And then the C-terminal part, the green domain, not enough is known about it, but it has sequence homology to citrate synthase. So that's why it's called a CS domain, citrate synthase domain. But as I'll show you later, actually, it's not really a citrate synthase. So work in Maria Fraser's group long time ago, they determined the structure of the N-terminal 820 residues of human ACLY. That's from 820 is right here. So everything is here. They did this by doing a proteolysis of full-length ACLY. So that gave them crystals. Full-length doesn't crystallize very well, so they couldn't work on it. So you can sort of see all the domains here, ATP GRAS domain, right here. Citrate domain, they have a citrate bond there even, and then CoA domain in the back, and then the histidine domain in the front in pink. So all 4 domains are here in that structure. However, this part, when you chop out the C-terminal part, the protein is a monomer. And the enzyme cannot bind CoA, cannot produce acetyl-CoA. So it's inactive in terms of the overall reaction. Okay? So why do we care about ACLY? Because a compound called ETC1002, Nesletol, I don't know how to pronounce that, but anyways, so this compound actually was approved 5 years ago for treating hypercholesterolemia. So a compound against ACLY actually is on the market for treating high cholesterol levels. And then another inhibitor, so this compound actually is a prodrug. It gets converted to a CoA ester. That's how I inhibit the enzyme. And another compound called compound 9 was developed by BMS, Bristol Myers Squibb, another pharmaceutical company in the US, back in 2007, shown like this is sulfonamide, biarosulfonamide compound. And that compound has 130 nanomolar IC50 against ACLY and has lipid-lowering activity in mice. And then also many cancer cells have upregulated ACLY activity. So ACLY is a target for various different things, metabolism or maybe even cancer. So that's why there was a lot of interest in ACLY over the past years. So again, this is the same company, Nimbus, developed this compound, NDI 91143. So it's also a sulfonamide biaryl, somewhat similar to the BMS compound shown right here, but also, of course, many, many chemical differences. And this compound, they did, again, kinetic assays, enzymatic assays. It shows compound is very potent, 7 nanomolar in their kinetic assay. And actually, it's competitive against substrate citrate. So it should bind where citrate binds. Simple. Very potent. And then non-competitive against ATP and CoA, the other 2 substrates. So now you know now how it works. And then, of course, this is later now, so you can, using modern technology, you can do a beer core binding assay using, against a small molecule, very weak signal, only 15 or so response unit, but that's sufficient to see a binding signal. You can sort of see touching in more and more compound. You see the on rate and off rate. From that, you get a KD, 2 nanomolar, similar to the kinetic assay. And then in my lab, so again, they reach out to me because of good history with them. So they reach out to me and say, okay, can you help us determine the structure of this compound bound to human ACLY? I said, sure, let's try it. So what we did first, we purify the protein, and then we do something called a thermal shift assay. Do you know about thermal shift assays? No? Okay. All right, let me explain that. So when enzyme, we talk about hydrophobic core, right? So when enzyme unfolds, the hydrophobic core gets exposed, right? So you have a fluorophore that binds hydrophobic residues. So before the protein unfolds, it doesn't bind very well. And then when the protein unfolds, it binds, and then the fluorescence will increase. And how do you unfold the protein? You heat it up. So you have all done real-time PCR. Well, you know about real-time PCR, right? You basically do PCR. So here, you use a real-time PCR machine. You do a temperature ramp. Every degree, you heat it 1 minute at that temperature, measure fluorescence. Heat down that degree, measure fluorescence. So that's what you're seeing here. Temperature in Celsius up to 90 degrees. And then over here is fluorescence unit arbitrarily. So they are scaled to 100. So you can imagine at low temperature, the enzyme is happy, doesn't bind the fluorophore. And then once it starts unfolding, it starts basically having a large increase in fluorescence. So that gives you a sense of stability of that protein. And then when you have a small molecule bound to the enzyme, you can stabilize or destabilize the protein. So you can change the melting temperature back and forth. If you stabilize the protein, they shift to the right, more stable. If you destabilize it, shift to the left, becomes less stable. Right? So let's look at the dashed line first. So this is N-terminal domain. Black dashed line is the N-terminal domain, the part that crystallized. So N-terminal domain alone actually is very unstable. The TM is only about 39 degrees or so, less than 40. But then when you have the compound at 91143, there is the red curve. You can see a dramatic shift by 5 or 6 degrees, which is very, very large. Typically, the thermal shift in the present compounds somewhere around 2 or 3 degrees. So this is a very large shift in the enzyme. And similarly, full-length enzyme, which is the filled black curve, which is right here, is more stable, 50-some degrees melting temperature. And then when you add citrate to it, it doesn't care. Citrate doesn't stabilize the enzyme. But when you add a compound to it, which is red curve right here, you see, again, a very large stabilization, 5 or 6 degrees stabilization. So that shows the compound binds to the enzyme, also stabilizes the enzyme. So this is another way you can test whether your compound binds. By measuring the shift of the TM, you can also get the KD out of it. You can do a binding assay based on this as well. So here we only just did one concentration to give you a sense of this dramatic stabilization of the enzyme. So anyway, long story short, we got the structure of the compound bound to ACLY and also ADP. So it turns out the part that crystallized is here. Compound, remember the enzyme, the tetramer? So the 4 colors, well, anyway, many different colors, but anyway. So this is the internal piece of each of the monomers, so 1, 2, 3, 4. So the 4 wings of the structure are the 4 pieces that were crystallized by Maria Friedger a long time ago. And then the C-terminal piece right here was in the center. That's actually the tetramer. The piece that crystallized actually do not really have much contact. That's why they are monomer when you crystallize them. And then the compound's bound right here in black. So you bind to the blue domain, domain, which is the citrate domain. So as kinetics shows, the compound binds to the citrate domain. That's where it competes against citrate. So if you overlay our structure of the full-length enzyme against the crystal structure of the internal piece alone up to 820, you can see overall actually it's quite similar here. Of course, this part is unique to the full-length enzyme. The CoA part actually is quite different. So you can see the compound sitting right here close to the CoA. So you could sort of imagine that's how it competes. Actually, it's more complicated than that, which I'll get to. So first, the middle part, which comes from the C-terminal domain of ACLY, that forms the tetramer. It turns out actually it has an active site. It binds CoA. We can also model in oxaloacetate. So essentially, the central region does have catalytic activity. It has an active site. It's a really beautiful tetramer right here. 4 different subunits coming together. But when we compare our structure of ACLY against citrate synthase, remember this domain has sequence homology to citrate synthase. So we align the structure of citrate synthase with our structure, citrate synthase in gray here. Everything aligns quite well, except this histidine right here, you see now replaced in citrate synthase in gray, replaced by phenylalanine in ACLY. Because of that, you'll see on the next slide, this histidine is critical for citrate synthase. So that means ACLY does not have citrate synthase activity. Instead, it's a different enzyme. We think actually it's a citric CoA lyase, the one that breaks down citric CoA, the last step of the reaction. So this is the reaction mechanism of citrate synthase. I won't go into detail in the interest of time. So essentially, histidine 274 is the critical base that basically critical acid that neutralize this part of the CoA. And this histidine is the one that's replaced by phenylalanine in ACLY. So that means ACLY cannot do the same catalysis as citrate synthase. Instead, it does the reverse reaction. So essentially, you can think about it. Oxaloacetate plus acetyl-CoA make you citric-CoA. The reverse will be citrate lyase. And then citrate synthase takes citrate plus CoA, makes citric-CoA. So that's the reaction. So they start from citric-CoA. One is the product, one is the substrate. So in this case, I have citric-CoA right here. And then this phenylalanine has really no direct role in the reaction anymore rather than the histidine. So now this aspartate is much important. So the histidine on the other side is the general base, withdraw the proton from the citrate, and then that causes everything to collapse. The lyase, this bond gets broken, and then this aspartate then protonates the leaving group. So that's sort of how the reaction goes. That means that the aspartate should be very important for the reaction. And so I already mentioned that. This histidine is critical for citrate synthase, but not important for CCLY. So when we mutate this aspartate to asparagine, we lost almost all the activity. And then when we mutate this histidine to alanine, we lost 4-fold activity. So I think the overall idea is the same. So this basically central region is a CCLY that catalyzes the last step of the conversion from citrate to acetyl-CoA. So last bit, how does the compound work? Just you saw earlier in haloxyphob, here you have a tremendous conferential change in the citrate domain before the compound can bind. So what you see here at the beginning, citrate right here. At the beginning of the issue, this arginine recognizing one of the carboxylate on citrate through ionic interactions. And then the arginine has to swing out of the way, and then everything has to open up, and then the compound can go in. So essentially, there is no binding site in the regular enzyme, in the native enzyme. Only through this very large conformational change, you can basically allow the compound to bind. And all these changes are illustrated on the left as well. The arginine essentially you can see swing from here all the way to the outside. So very dramatic conformational changes. So then if you look at the cavity, I guess This is done using PIMO. If you don't know how to do it, I guess we'll get to that as well. If you look at this enzyme, in the complex, the inhibitor, there's a large cavity where the compound binds. And if you look at the enzyme bound to citrate, there is no cavity. So even though I showed the compound here, the compound is actually clashing into the enzyme. For example, this arginine that's recognizing the citrate, that they are directly facing each other, and the arginine directly goes into the compound. So there's no way for the compound to bind. Arginine had to swing out of the way, everything had to open up, and the compound slot in, and citrate goes away. So that's how it competes. A conformational change in the enzyme that kicked out citrate. So you can see the compound actually doesn't really overlap the citrate directly that much. The competition is mostly indirect, but still, it gives you the, okay, so now you can make a structure like this, a binding mode and everything, how the compound sits in there. And then Nimbus has a very strong collaboration with Schrödinger, which is one of the leading companies for doing computational chemistry, software. They also did the docking for ND6, the sulfur binding site docking. They did that as well. So they can do free energy perturbation calculations using the binding mode. And then they have a series of different compounds. They know their experimental KD, which can be converted to a delta G based on the KD. And they can calculate the delta G for this compound based on free energy perturbation. You can see a very good correlation between observed KD or delta G and calculated delta G based on this binding mode. So providing additional support for this observed binding mode for this compound. So this is to give you another story of how you can develop some molecules using, essentially, in this case, a screen, if you will, or a lead, and then using structural biology to guide you to do better compounds and everything else. So many other companies are also looking at ACE1 inhibitors, and I'm sure many other binding modes will show up as well in the near future. So the summary, I guess I'll skip over. I already said most of these things. I'll skip over. And with that, I guess we have a few more minutes. So if you have questions or whatever you'd like to discuss, we can discuss as well. Yeah.
Is it possible to predict the conformational change by computationally when some molecule is binding to the protein or protein complex?
Which conformational change?
Like, suppose a molecule is binding to a protein and it's leading to a conformational change in the protein, or protein complex.
Mm-hmm.
How we can predict the conformational change happening computationally without?
Okay. Very good question. How can you predict conformational change? The answer is, I don't know. I wish I know. I wish I knew. But I guess the idea essentially is, so far, what you see here has been purely discovery driven. You have a small molecule as a probe, you hit it, and you see a conformational change, and things happen. And I guess when you look at protein structures, even without ligand bound, you do see different states in crystallography. Like in a crystal structure, oftentimes you have 2 or 3 molecules that are unique, and they're a little bit different. So you can sort of see that's conformational dynamics in your protein, but maybe not so dramatic as you see here. So these probably you can think about the conformational sampling, state A, state B. So the percent of A and B could be very, very different. So in the absent compound, mostly A is there, B is a little bit. In the present compound, it goes this way. So it's going to be very hard to crystallize this on its own. You need something to stabilize it. But that's doing experiment. If you do simulations and this and that, you might be able to observe something like this, although you probably have to do very, very long simulations to see something like that. And the other thing that you can do is AlphaFold. So sometimes AlphaFold, though you say AlphaFold only gives you one structure, actually it's not really true. Sometimes many cases you have active site doing this, right? Open and close. Sometimes give you a closed state, sometimes give you open state. How it does it, I don't understand, but I have certainly seen that. I've seen that in our crystal structure is closed, but AlphaFold somehow pried it open, put something in there. How does it know? I don't know. So that may be another way to see that. But yeah, it's a very good question. That very, very powerful. As I mentioned, it happens quite often these days that you have the active site region of the enzyme that's conformational dynamic. And this dynamic opens a little pocket, the compound goes in there, the enzyme's dead. I'll show you another example for protein kinases later, I guess on Friday. Yeah, very good. Any other? Okay, then I guess finish right on time. So I'll see you. Sorry. I guess Professor Shukla just came in. I think we'll take a 10 minutes break, and then we'll start again. Okay, 10 or 15. I don't remember what he said. Whatever he says. Okay, we'll take a break for now. So we'll continue at 12:15.